Thanks for a kind introduction. Good afternoon, everyone. And somehow, I guess, in some way, I will follow up on Arjun's talk. The topic is about the same. It's the use of near-infrared light to probe deep tissues, right? And since I'm following up on him, but I didn't want to actually overlap on everything that he was saying. So I'll present one part of what we do in our lab, which is basically focused on neuroscience. So that's going to be more of a neuroscience talk than an optics talk, actually. So since I have such a short time, I just want to make sure that I have the most important slides first. So, and the most important for me is actually to acknowledge all the work that I'll present here is based on all this group of people, this wonderful group of people that actually I was fortunate enough to gather together in Campinas, right? So basically, we are at the University of Campinas. Campinas is a small town, well, not that small actually, about 1.5 million, really northwest of São Paulo. And the university has a somewhat large campus. For those of you, some of you will get the opportunity to know the university tomorrow, and then you'll be able to, well, have a feeling of taste exactly what the university is. So, and we have the biomedical optics lab there. Basically, we try to develop diffuse optical instrumentation for mostly neuroscience applications, brain in general, right? So you can actually see and check our website. It's that.light4brain.com. Or if you like Facebook, you can actually like our page on Facebook. It's LOB, that stands for Biomedical Optics Lab in Portuguese, Unicamp. So basically what we do there, we are in a physics institute, and what we do there is study scattering, right? So this is one of the very first experiments we've done over 10 years ago. And with single scattering that Arjun already analyzed. The experiments we do is actually based on this phenomenon, but it's much more complicated. It's not even single scattering, it's multiple scattering, as Arjun mentioned, right? But to understand a little bit of the basics, and I'll be a little bit repetitive in this beginning, I just want to start with a simpler problem that we know mainly from chemistry, right? So if you shine light into a medium, and this medium absorbs light, right? So you have Beer-Lambert law, the light decreases exponentially as it actually goes through the medium. And basically the rate of decrease of light is proportional to the absorption coefficient, which is related to which molecules you have absorbing in this medium, right? So by measuring the intensity of light, you can actually get an idea or estimate the concentration of the molecules that actually absorb light. In tissue, in the near-infrared region, there are basically two main molecules that absorb light, oxy and deoxyhemoglobin. They're both in the brain, as we already heard, right? They have different optical signatures in the near-infrared from 700 to 900 nanometers. So you can actually use that if you have at least two different wavelengths to estimate the concentration of oxy and deoxyhemoglobin in tissue, right? The problem, as Arjun already pointed out, is basically we have scattering in tissue. It's not only absorption. And scattering, it turns out that scattering in tissue is very high due to the heterogeneity that we have in tissue. And we know how to deal with the problem, right? We just learned about that in the past 2 days. It's diffuse. We have a diffusion equation that can deal with that. The problem is, if you shine continuous wave light, you cannot separate absorption from scattering, right? So the intensity for continuous wave illumination, the intensity is proportional to both absorption and scattering, and you don't have enough information to separate absorption from scattering. So what we do is basically, if you want to shine continuous wave light, you can, because of high scattering, light diffuses. Part of this light comes back to the same surface. And since we cannot separate absorption from scattering, we cannot at least monitoring the intensity of light over time to calculate the changes in the optical properties, right? And under certain assumptions, there are reasonable assumptions for neuroscience in general, that scattering doesn't change much. That's not always true, but it's true most of the times. You can attribute all the changes that you have in light to changes in absorption, right? So basically, if you monitor light intensity over time, you can compare the changes in the absorption coefficient over time. And if you do that with two different wavelengths, you can actually extract and calculate the changes in oxyhemoglobin and deoxyhemoglobin concentration in tissue, right? So that's actually very useful. It turns out that it's a simpler model, but it's very useful mainly in neuroscience. And I'll just go through, since this is a lecture, not a course, I'll just want to show you some of the recent studies that we've been doing with this technique, very simple, particularly in neuroscience, right? So one big field of neuroscience is called functional neuroscience, and the idea basically is we can always perform a task, and this task can be, we can compare the changes in the brain that are correlated with this task compared to a baseline, right? So that's a functional protocol. You perform a function, right? And you can compare, and you can repeat this function over time, and you can compare what happens in the brain or different regions of the brain during this task with the pattern that we had on baseline. So in a very simple way, this movie actually represents what a functional experiment performs, what a functional experiment is exactly. For a finger tapping, like if I actually if I tap and do a motor experiment, that's related to changes specifically in one region of the brain, the motor cortex. So basically, that's more or less the idea that we have, right? So you can actually have light sources and detectors. You can monitor the changes. And if you perform a task, a finger tap in the right hand, you see a change in your left hemisphere around the motor cortex, right? If you stop, the changes disappear. They go back to what they were in the beginning. And then if you start tapping your left hand, the right hemisphere in the region of the motor cortex changes, right? So basically, performing these tasks over and over again, you can always compare what the change in the optical absorption and therefore the change in hemoglobin concentrations are with the baseline that we had. So that's the idea in functional neuroscience, and that happens, physiologically speaking, because when we have an external stimulation, you have, your neurons, all the cells that you have in the brain, they also will perform a task, right? So they will basically have more activity, and this activity needs energy. And it turns out that in the brain, we don't have a storage of energy. So all this energy comes from a very complicated network of blood. And the energy, O2, basically, and glucose comes from the blood to actually supply the demand in neuron activation, right? So there's a relationship between an external stimulation that actually induces a neuronal change and the hemodynamic change, or basically the blood. You have an increase most of the times in blood volume, in blood flow, and therefore if you have more blood there, you have more oxyhemoglobin coming up. And because the neuronal is using this energy, you have less deoxyhemoglobin there. So basically, that's the main idea. You have the cell working, and basically the vasculature bringing blood and all the molecules that you need to supply the demanding energy. One very simple and direct way to do this is an experiment that we did with rats a long time ago, measuring electrophysiology, so the activity due to the neural cells there, during a very simple protocol, which is a whisker stimulation in rats, right? So you have the whisker in rats, you can actually just tweak or tilt this stimulation, and basically there is one region on the brain of the rat that is responsible to control this whisker activity. And as soon as you do that for 2 seconds at 8 hertz, that's what we did here, you see the spikes in the electrophysiology, and that's correlated with the change in hemodynamics. So in this case, we measure an increase in blood flow, an increase in oxyhemoglobin, a decrease in deoxyhemoglobin. And if you sum oxy and deoxyhemoglobin, you have a change in total hemoglobin that's proportional to blood volume in that region, right? So basically, the idea is actually electrophysiology drives flow during a task. And that's all happening to supply the demand in metabolism, right? And one of the nice things that you can do with this is actually you can model the brain. I'm not going into details of this. David Bowles will talk a little bit more about this next week. But what you can do is model what's going on from the electroactivity to the hemodynamic change. There's a lot going on in terms of metabolism, in terms of arterial, so the vessels are dilating or constricting to control blood flow there. And you can actually just model predict this by measuring both electrophysiology and optically the region due to a single task, right? So that's one of the things that you can do, understand basic, fundamental neurophysiology behind the tasks and activation of the brain. It turns out that although it's been centuries, we don't really fully understand all these relationships. We know that there is a relationship between the changes in electrophysiology and the changes in metabolism, but we don't understand the full details of it. We don't understand if it's nonlinear or linear relationship, and if it's a nonlinear, we don't understand the degree of nonlinearity of this, meaning that if we, for example, if we double the activity here, are we actually doubling the amount of blood flow being delivered there? So that's a very simple question if you want to understand the systems, and we don't have a definite answer for that yet. And all the functional connections of the brain are being organized. But unfortunately, we don't have many opportunities to measure babies, right? You're not allowed, for example, to put a baby inside an MRI, which is the traditional way to do neuroscience, at least not a healthy baby, right? Because you got to anesthetize the baby. So you better have a great argument to put an anesthetized healthy baby inside the MRI. With optics, it's completely non-invasive. There's a lot of motion artifacts, and you have to take care of that. You have to deal with it. But the fact is, you can measure babies. They don't need to be anesthetized. You can just put the probes in, like we did here, right? And some of these fiber optics bring light. Some of them collect light. And by just measuring the amount of light at 2 different wavelengths, we can actually estimate oxy and deoxyhemoglobin change. This is one very early study that we did recently in babies. And the idea here, we were actually interested in 2 things. First, comparing preterms to full-term babies, right? So it's well known in psychology and also in pediatrics that preterms have a delay in cognitive and, well, executive functions too. And so we wanted to actually compare preterm babies on the left side with full-term babies on the left side when they were performing a motor task stimulation, right? So we would actually just stimulate the motor performance of them with their hands. And what you can see is basically the change in the motor cortex due to this very simple task in babies with 6-month-old, okay? And there is immediately a change, right? Because For preterms, you see a bilateral activation, so both hemispheres actually light up, meaning that they both are activated for this task during the preterms, but not the full terms. And the idea here is actually you are seeing basically a specialization effect. So the brain in the preterms, in this case, are not fully organized and not fully developed. So that's why you need a lot of regions being recruited to actually control this task, which doesn't happen with the full term. And that's actually corroborated with measurements at 1 year old in these babies, right? Because over time, this happens, and that's known in psychology and functional neuroscience as specialization, right? So the brain, different regions of the brain get specialized over time, right? And in this case, they got specialized around the motor cortex, right? If you actually look at, for example, the regions that are activated, you see the same profile that I showed you before, an increase in oxyhemoglobin, a decrease in the oxyhemoglobin, both, all different regions here, right? So this is one example that what you can do in functional neuroscience with diffuse optics or near-infrared spectroscopy that people call also as NIRS or F-NIRS, from functional NIRS, right? Another thing that you can do, and that's also very hard to get from any other technique, but you can do it with optics, is during physical exercises, right? It's really hard to get someone inside an MRI, for example, to perform physical exercise, right? You need the person to be rested and not moving that much. So here, what we did, that's an ongoing study, and we were interested really in looking what the brain does when a person performs a strength exercise, in this case, a dumbbell concentration curve, right? And there's a lot of debate, and mainly in the physical exercise area, asking what really happens in the brain or in your muscle that makes you stop an activity, right? So if you are running and you stop, what really happens that you made the decision to stop, right? So the traditional theory says that, well, maybe your muscle saturated and you don't have enough energy to keep going. Some more modern theories say that basically your brain made the decision to stop even if your muscle didn't actually saturate, right? So in this sense, we were actually trying to see what happens in the brain during a very heavy task, right? So for example, you can see here the motor cortex associated with the movement, but also there's a lot of activity in the prefrontal cortex in this case. And when I say activity, what I'm always referring to is an increase in oxyhemoglobin, a decrease in the oxyhemoglobin that is correlated with the task, right? Temporally correlated with the task. So what you can see actually is if you look at the amount of regions that actually are activated during the task, the motor region doesn't really change much if you do a very heavy weight here or a very low weight here. But the prefrontal cortex does have a change, and it depends literally on the amount of weight that you are able to perform this exercise, right? And the lower the weight, the more regions in the prefrontal cortex you actually see an activation, right? So it turns, and the prefrontal cortex is the region of the brain responsible to decision making, among other things, right? So it appears we are just doing that. That's still a study ongoing. That's preliminary data, but it appears that the brain actually plays an important role, mainly for repetitive tasks. Another example that you can use is NIRS is on cognitive tasks, right? So in this sense, in this experiment, we're trying to see what happens in the brain when someone reads a passage, right? It's a small passage. And you can compare the reading period with the rest period, and you see that there's a lot in the brain going on when someone is reading, right? There are regions in the brain that's related to reading, and we know that from neuroscience, basic neuroscience. There are also regions that are related to emotions during reading, right? And one interesting question that we did in terms of methods is, does it make any difference if you're reading silent, if you just read a passage silently and if you read out loud, right? Because that would induce, or that could tell you that there is an influence of motion artifacts in the data, right? So what you can see here is actually, basically, you don't have any change if you read aloud or if you're reading silence, which actually strongly suggests that what you're seeing is basically an activation due to reading and not any activation due to any potential motion artifacts, right? In all these regions, the different regions here, you can see also an increase in oxyhemoglobin, a decrease in deoxyhemoglobin, and they turn out, even the dynamics of reading is about the same in terms of activity, right? So there's not really a big change, right? As Arjun already pointed out earlier in his talk, we got to be careful with a lot of things, right? So one of them is the extracortical contributions to the optical signal, because when light goes through, it actually probes the cortex, and that's what we are interested in neuroscience, but it also probes a lot of extracortical vessels, right? So, and they may change depending on the task that you are doing. They may change, and their changes also can also be correlated with the task. Just to give you an example, or two different examples. The exercise, the physical exercise, the dumbbell exercise that I told you before, right? If you don't properly correct and remove the extracortical contributions, you don't see the activation channels, right? So these two curves are oxy and deoxyhemoglobin in your raw signal without really properly accounting for extracortical contribution, and you basically see no change at all. And once you remove the cortical contributions, then you see the increase in oxy and the decrease in deoxyhemoglobin, totally correlated to the task, right? So if you don't do it correctly, you would actually think that there's nothing there, there's no region activated associated with the task that you are doing, right? And this happens because if you're performing kind of a tiring task, your heart rate goes up, your blood pressure goes up, and therefore you have a lot of changes in blood, right? In the blood volume. But they are systematic changes. They change across the whole body, not only in the brain cortex, right? So that's why you have to account for that. Another example of that is with a finger tapping, right? The very simple finger tapping that I told you before. This is not in babies, this is in adults. And you can see that basically, if you don't remove the extracortical contributions, right? Putting a short channel, as also Arjun mentioned to you before, basically you see a lot of changes, but they are not realistic changes. They are just changes in the extracortical region, not really changes in the brain. So if you, and after you account for the extracortical changes, then you see what you'd expect for a finger tapping experiment, which is a change mostly located in the contralateral hemisphere, right? So in this sense, it was a right-hand task, motor task. You see a response in your motor cortex on the left hemisphere. Right? So that's to say that it's very useful, but you really need to know what you are doing, otherwise you may not see whatever you're seeing, and usually we tend to blame the technique and say that the technique doesn't work, which in fact, that's not the case, right? It doesn't mean that we don't have problems with the technique, right? So this is one very simple question that we've been asking ourselves in our lab recently. What happens if you perform a task, the same subject, 3 different times at 3 different days, right? So this is an example of a finger tapping performed by one subject at 3 different days. Same subject, same task, during the same time of the day, but different days, right? And if you see, there is a lot of intra-subject variability, right? So sometimes, or most of the times, you see what you are supposed to see, but you see a lot of things that should not be there. There's variability there. And you can actually go even further and measure the same thing, not 3 days in a row, but the same day at 3 different periods of the day, right? So you perform the same experiment with the subject in the morning, in the afternoon, during the night, and you see completely different patterns. So there is variability there. If you basically look at that as a whole, the region related to the task is always there, right? This is a frequency map showing basically the amount of times that that region showed an activation, right? So for a finger tap on the left hand, you would expect the motor cortex on the right hand to be activated, and it is there all the time, but there's a lot of variability around all the other regions. And that's due to a lot of things. Your systemic physiology changes during the day, right? And it also, of course, changes across different days. And that's part of it. There is a very low signal-to-noise ratio in the data. We are always measuring these things at the limit of your noise. So there's systematic errors, and that leads to variability. And that may be an issue if what you are trying to do is monitoring, longitudinally speaking, monitoring subjects in a longitudinal way, right? So if you want to, for example, monitor a treatment over days or months, right, this variability plays an important role. So you may see changes, but they are not really real changes related to the treatment. They are just variability across the days, right? So that's something that actually poses one of the limitations on the use of NIRS for neuroscience currently, right? But there is also another set of stuff that you can do with optical methods in neuroscience and basically with NIRS in neuroscience, right? So everything that I've been doing is on functional neuroscience, which means that someone is performing a task, and I'm just looking at the changes that are correlated with the task. However, the brain, even if it's not performing a task, it spends energy, right? So that's well known and well documented since early in this century, right? So we can actually even estimate the amount of energy usage by the neuron cells and the glial cells. So even at rest, when we are not doing supposedly anything, there is energy consumption, which means that there are changes and spontaneous changes in hemodynamics, in blood flow, right? And this was actually first observed back in 1995 in MRI, in an MRI context by one group in England. And what they saw was basically, well, if you do finger tapping, and that's kind of the base experiments that we want to do in neuroscience if you're not used to neuroscience, just do finger tapping to make sure that you know, because that's that's supposedly a very repetitive task, which I just showed you that it's not like that, but that's what people believe in neuroscience. So if you do a finger tapping experiment, you see that there is a change in the motor cortex, right? And if you do in one hand, there is on the contralateral hemisphere. If you do the other hand, there is the contralateral hemisphere. However, if you just don't do anything and just collect data while the subject is not performing in any task specifically, but if you compare the time series, the changes, in this case in the MRI intensity, right, during rest, with the time series with all the other channels, you see that basically the channels that are mostly correlated, if you pick up, for example, a time series in the motor cortex, the channels that are mostly correlated with that time series is exactly the other cortex, right? So just by comparing specific regions of the brain at rest, you can get profiles that are very close to what you would get during the task, right? So we know from this kind of open a big field in neuroscience, mostly in MRI, and basically people can identify at least 13 different, they call it functional networks, which is basically regions that are closely related to regions, closely related functionally speaking, right? So they are functional responsible for the same tasks. And you get one of these networks depending on where you pick one of the regions to actually look and compare with all the other regions, right? So that's well known in MRI. You can actually do that with optics too, right? So there's a lot of people doing that with optics only. And this is one experiment that we did comparing basically the networks that we get with MRI and with optics. So we did a multimodal experiment. We put all the light sources and detectors inside the MRI scanner, and we collected data during rest, right, when the subject was not performing any task, with both techniques at the same time, right? So, and you can see that there is, there's a change in spatial resolution. MRI has a better spatial resolution than NIRS, but what you can see actually, briefly, you have roughly the same networks that you find with MRI, you can also find with NIRS. I'm just showing here 3 different networks related to the visual stimulation and auditory, right? You can actually go ahead, because since you are comparing different regions, right, you are assuming there is an interaction with the different regions, right? So, and there's a whole field on physics related to interactions, right, of particles in general. And one of way to use that, one of the approaches is to use graphs, right? So what you can do is just collect data, right, the intensity at different regions of the brain, and then you have, over time, just spontaneous fluctuations because there's no activity, there's no task related to that. And what you can do is actually just pick one of the intensities that are recorded and compare to all of the others, and you can do that for all the intensities that you recorded, right? And then by comparison, you can just, for example, taking the Pearson correlation coefficient, right? And basically, when you do that, there's one way to see if these spontaneous changes are correlated in time or not, right? So, and you can set up a threshold in this correlation coefficient, for example, and just assign 1 if that correlation between 2 different regions of the brain is above your threshold, and 0 if it's not. So that will give you a profile of a graph, basically. That's basically saying which regions of the brain are actually connected to each other, right? And once you have this, you can actually compare and quantify your graph. So you can quantify, for example, the number of links, the number of connections that you have in your network. You can quantify for each region of the brain the average number that they are connected, how they are organized, how they are clustered. And basically, if you do that, and if you compare the optical data with the both data, you see virtually the same profile of the task. I'm plotting here all these numbers as function of the threshold, right? So the threshold basically here is arbitrary, so I can pick a threshold of 0.5, right, for the correlation coefficient, or 0.3 or 0.7. There's no reason I can choose one or another. So what we do is actually we look at all of them, right? This is, for physicists here, this is kind of my order parameter of my system, right? So basically, if you look at dynamics, independent of the threshold, you see a very similar profile with the different techniques being optical or MRI, right? However, again, there is variability, so we can ask the same question. If we put the subject there and we collect data at 4 different time intervals, right, during the same day, and do all this exercise, you see completely different graphs, right? So there is variability even in the resting state. What is interesting for us is actually, regardless of the variability here, the metrics that you have for quantifying these networks, these graphs, they basically are the same, right? So regardless of the variability that you have in the graph, right? The properties of that graph remain stable over time, right? So we think that that's actually a good way to compare subjects over time in the periods of months or the periods of days, right? Because not by their graphs or by, but also not with their graphs, but with the properties that you can quantify from the graphs, right? Again, for the physicists here, one way to interpret that, right, or my way to interpret that is actually we have different microstates of the brain, right? They all are on the same macro state, right? They all lead to the same region, right? But there are different ways that you can organize these connections using the same profile or the same macroscopic profile, right? And once you do that, you say, well, visually you have the same profile for the person, it's very stable. The question is it the same for everyone or is it different for everyone, right? So that's a very simple question that you can ask too, right? You can measure a profile of the networks, and you can do that for a lot of subjects, and you have a database of subjects. And the question is, given one unknown subject, can I recognize which subject I'm talking about, right? It's kind of like another way to pose this question is the resting state profile a fingerprint of one specific unique subject, right? So one way to do that is actually just compare that with the database that you have and just looking at the similarities, right? Just looking at the similarities of the profiles of the connections, you can see which subject in your database is more similar or is most similar to the profile you run the test, right? So if you do that in a leave one out approach, which is basically the same idea that we heard before, we have, in this case, about 30 subjects. You just do it, you train that with one set of subjects, and then you do that, and you're always testing with the one that you left out, right? And if you do that, and you actually switch the subjects across the time and run that out again, you have about 90% accuracy, right? So just given one profile, I can tell which person it was, basically on their functional connections measured with optics, right? So in order to finish and wrap up, I just wanted to actually give you a flavor that NIRS, or near-infrared spectroscopy, is a very useful tool in neuroscience. It can be used from babies to adults. It can be used to probe tasks like physical exercise to actually cognition. And you can also use that in real environments and not inside an MRI, for example, machine, right? You actually can help us understand a little bit of neurophysiology, right? So if you combine particularly optical methods with electrophysiology, you can understand what's going on with all the processes. Not all, but at least it can shed light on what's going on, right? However, it's not plug and play, right? You gotta know what you are doing. There are a lot of issues that you have to understand in order to really get the profiles, right? So there are motion artifacts that I briefly mentioned here, extracortical contributions that I showed you here. There's data variability, inter-variability, inter-subject variability, intra-subject variability, right? But despite all that, I truly believe that resting state functional connectivity with optical methods can be very useful, mainly for brain diagnosis, right? So with that, I want to finish, and thank you for your attention.
Questions?
Rickson, I just wanted to follow up about the nonlinear response. So what exactly does that, what's the experiment to do?
Well, so
like, oh, you explain.
Yeah, what people try to do is actually, if you perform a task, so, I don't know, perform any task, and you see the amount of change in blood flow, for example, right? And the question is, for example, if I do finger tapping, I can do that for 5 seconds, and I see an amount of change in oxyhemoglobin. if I double the time, if I perform the finger tapping for 10 seconds, is the change double too? Right? So if I perform for 5 seconds and I see a change of 5 micromolar in oxyhemoglobin concentration, that means that if I perform the task for 10 seconds, the change will be 10 micromolar?
But it would be a change of 5, but just for longer, right? I thought it was like.
Well, so that's the question. It really depends. So we don't have a definite answer for that as far as I'm aware. With duration or intensity, if what you're varying is actually the duration of the task or the intensity of the task, the system is quite linear. That means that if you double the time, you will double the peak, the amount, right? Or the total integrated response. Yeah. You can, well, also the peak. The peak actually goes linearly. The peak, right? It can peak up and then goes back to a different level. But it peaks higher, and it peaks about roughly double if you double the duration of the task, right? But with frequency, so if I kind of do finger tap at 1 hertz, and I get a response, but if I do that at 3 hertz, that is highly nonlinear, right? And we don't understand why. That's the main idea.
So, could psychological stimulation during finger tapping or a task, functional task, change the levels of oxy and deoxyhemoglobin, or these levels?
I mean, if any task will change the levels of?
Yeah, see if psychological stimulation during the task would differentiate the levels.
I'm not sure I understand your question, but yes, and this, if you do a psychological stimulation, it will temporarily change the task. The question that we don't know is if this change can be permanent or not, right? So that's a big question, and all the studies that I've seen show that it's just temporary. At some point, if you stop the stimulation, it will go back to the baseline. It may take longer, but it will go back eventually.